Second. Decoding marine records of past global warming events using environmental magnetism.
All right, thanks, Rob. And thank you very much for having me here. I'm excited to share with you today how we can use environmental magnetism to decode past records of global warming events in marine sediment cores and start to answer questions about how these events affect changes in the environment, including changes in the iron cycle. And I'm going to show you two examples from two marine sediment cores. One from Wilson Lake, New Jersey, that records a Paleocene-Eocene thermal maximum, which Nono mentioned yesterday. And then the other example will be from the Holland Ballpark Core in present-day Virginia, which records Oceanic Anoxic Event 2 from the Cenomanian Turonian. And Tim actually briefly mentioned that last night. And before I dive in, just want to give a shout out to all the individuals and funding agencies listed here. Science really is a team effort, and without all this support, the science I'm about to show you would not be what it is today. First, a quick primer on what magnetofossils are and why we care about them, because I'll be talking a lot about them. Firstly, they're the fossil remains of iron biomineralizing organisms, and this includes magnetotactic bacteria. And as Andrew mentioned yesterday, molecular clockwork from Wei Lin and others suggests they might have even been around since the Archean. Due to the stable single domain nature of most magnetofossils, they can contribute a large proportion to the natural remnant magnetization of sediments. And because of the environmental sensitivity of the organisms that make magnetofossils, they can be used to study environmental change throughout the geologic record, particularly the iron cycle. So because of the biomineralization, fossilization, and dissolution, whatever the fate of magnetofossils, they're directly contributing to the iron cycle. And there's two general categories of magnetofossils, the first of which is on the left here. These are conventional magnetofossils. So these are the remains of magnetotactic bacteria, which again are still around today. And they're super tiny, about 20 to 120 nanometers. And they come in a variety of shapes and sizes or magnetofossil disparities. So I'm showing some examples of that on the bottom left here. But notably, they can be kind of split into two categories, these more equant ones. That have a smaller shape anisotropy, so they have a lower magnetic coercivity. We call that biogenic soft, and we represent that by the equant cartoons up there. And then some of them are more elongated, so they have a slightly higher shape anisotropy, slightly higher magnetic coercivities. And we call those biogenic hard, and that's represented by a more elongated magnetofossil cartoon up there. And these differences in shapes kind of roughly correlate to different environmental conditions. Next, on the right-hand side here, we have giant magnetofossils. So per their name, they're much larger than conventional magnetofossils. They're usually greater than 1 micron in size. But unlike conventional magnetofossils, we have no modern analog for these. We have no idea what made them yet. We've only found them during now Cretaceous and Paleogene warm periods. So the last, or about 94 to 55 million years ago. But because of that, we think that they're related to some of the environmental conditions that were exacerbated during these warm periods, maybe even the iron cycle, so increased iron from increased runoff and weathering of continental environments. But luckily, we do have the biogenic needle signature, which fingerprints these very elongated magnetofossils with an even higher shape anisotropy. So they have a much higher magnetic coercivity, and we can distinguish these in using magnetic measurements as well. But there are also different shapes and sizes or disparity of giant magnetofossils. So we also have spearheads on the top right here. We have giant bullets, and we have spindles. Now, what I want you to take away from all this is we have, again, all these different shapes and sizes or magnetofossil disparity, and they can roughly correlate to different environmental conditions. And conveniently, we have these three categories of magnetofossils that can be remotely sensed in bulk sediments using environmental magnetism in the lab. So I'm going to show you exactly that, how we use environmental magnetism to distinguish between the biogenic soft, biogenic hard, and biogenic needle components, and how we can use that to begin to interpret environmental change. So for those that weren't at the fork workshop yesterday, here's your quick primer on what first order reversal curve data sets are, which are our tools of choice. And that's because they allow us to probe the magnetization state of different magnetic assemblages within a bulk sediment sample. So we can remotely sense different magnetic assemblages within a little rock chip without having to break it apart or do more advanced extraction techniques. And quick primer on how to read a resulting fork diagram. Across the x-axis is magnetic coercivity or hardness. Across the y-axis is our magnetic interaction. And then on the far right, our color scale is the amplitude or signal, which actually corresponds to the change in slope between the individual first order reversal curves in the raw data. What you might notice on this particular fork diagram is this area of high signal across where y equals 0, where interaction equals 0. We call this the central ridge, and this is a fingerprint when combined with this negative region in the bottom left of single domain magnetite. And in this case, we confirmed that this single domain magnetite is actually biogenic from magnetofossils. We can take it a step further, like Ramon was discussing yesterday, we can look at the contributions of magnetofossils to the central ridge and basically do a cross section across the central ridge. And that's this red line here, which we call a coercivity distribution. And here's where those magnetofossil components kind of roughly fall on that distribution. So on the bottom left, again, our more aqueous particles are the most magnetically soft, so biogenic soft. And then in the middle here are the second category of conventional magnetofossils that are slightly more elongated. So they have a slightly higher magnetic coercivity. And then on the far right, for the highest magnetic coercivity are biogenic needles, which are a type of giant magnetofossil. And as No went into length yesterday, we can actually, oh no. Is that better? Oh, I can give you that too. All right. You'll know I'm talking. Thank you. And as Nona went into length about yesterday, we can actually use statistics to begin to get quantitative estimates on the different proportions of these magnetofossil assemblages within sediments and actually get time series of this. So for those that were here yesterday, I hope this is very familiar and you're able to reproduce something similar. This is a result of principal component analysis on 65 different first order reversal curve data sets. And I'm going to show you the record that it came from later, but let's just focus on the analysis for now. Then the bottom left is our actual principal component analysis, our score plot. So we have two principal components describing the variation in the data. And then across the top there are our simulated end members. So this is what our analysis came up with on its own. If we zoom in, what I want to highlight is that these simulated magnetofossil end members from our analysis are strikingly similar to what we would expect for real data sets. So just to run through them, over here on the left is end member 1, and it has the highest contributions to the central ridge, high coercivities. This is fingerprint of our biogenic needle component. In the middle, in green, is end member 3, and this kind of has the most mid-range coercivities in the central ridge area. And this corresponds to our slightly more elongated conventional magnetofossils or the biogenic hard component. And then on the right in blue is end member 2, which has contributions to the central ridge close to the origin there, with the lowest coercivities. And this corresponds to our more equant conventional magnetofossils or the biogenic soft component. And if you can't see that, it's hard to see the contributions to the central ridge, just take a look over here in the top right at the coercivity distributions across the central ridge. Just to run through those, on the far left of the plot here, we have end member 2 again in blue, with a peak in the distribution at lowest coercivity. So those are our equant magnetofossils, biogenic soft. Next, over to the right in green are those mid-range coercivities from our biogenic hard component, or end member 3 in this case. And then finally, with the highest coercivity and largest distribution across the central ridge is end member 1, which is our biogenic needles. And again, Nona mentioned that we can look at these changes throughout time series. So this ternary diagram here is showing you that we have these different end members in our analysis, which we've interpreted as these different magnetofossil components. And it's color coded from up core to down core with the different shades of white to black. And this is showing us we have differing proportions of magnetofossils through these 65 data sets, so the stratigraphic interval in this particular core. So next, I'm going to show you how we can use that information through time to start to interpret environmental changes. And again, this example is from Wilson Lake, New Jersey. And what's great about Wilson Lake is that it's a marine sediment core that records a continental shelf environment over the Paleocene-Eocene thermal maximum, which is a global warming event that happened about 56 million years ago. And it's not perfect, but it's a really good analog for modern climate change. Just to place ourselves in time real quick, with this figure, we're going from about 250 million years ago in the top left toward present in the top right. And this black curve across here is delta 13C carbon isotopes, telling us we have changes in the carbon cycle over this time. And in red is our reconstructed temperature. And so at 56 million years ago, in this red box is the PETM, Paleocene-Eocene thermal maximum. And you'll notice we have a negative carbon isotope excursion with an increase in global temperature. So going back, again, we have this 4PCA analysis that gave us magnetofossil proportions for 65 data sets through this marine sediment core. So what we're going to look at here are those individual proportions plotted through time in the Wilson Lake core. So this red dashed line at the bottom is the start of the PETM, and we're going forward in time as we go up until we reach that squiggly line, which represents an unconformity where our data gets chopped off. And in purple here to start are proportions for N number 1, which again is our biogenic needle component. And where we see peaks in this magnetofossil N number, we interpret there to be a sudden increase in coastal runoff and perhaps iron supply. Then in the middle here in blue, we have N number 2, or our biogenic soft component, those equant magnetofossils. And where this N number is greatest, we interpret there to be more stratification at the seafloor. So there is less nutrients and oxygen making it to the ocean floor. And then finally, we have N number 3 in green on the far right. And this represents our biogenic hard magnetofossils, slightly more elongated ones. And where we see peaks in this N number, we interpret there to be an increase in seasonality, specifically seasonal nutrient cycling or seasonal eutrophication. And I want to just highlight here that these interpretations didn't come from thin air. So we do have a handful of these studies in the modern, and very recently we actually compared them to benthic forminifera to kind of nail down some of our interpretations. And here is exactly what we did. So for those that don't know what benthic forminifera are, they're microbes that make calcareous shells or tests. And similar to magnetofossils, you can split them into different species associations that have preferred living environments in the water column. And compared to magnetofossils, they are much more well-studied and accepted paleo environmental proxies. So this is exciting because it's the first time we've compared magnetofossils to other organisms. And we did that by comparing our magnetofossil N member assemblages to these species associations from the benthic forminifera. And I'm showing that how we did that using non-metric multidimensional scaling in the bottom right. And the take home here is that, yes, these different species associations correlate nicely with changes in our magnetofossils. And perhaps even more exciting is that the magnetofossils picked up from some of the finer changes in the environment than the benthic forams did. And they are even preserved in these intervals that are affected by carbonate dissolution where the forams end up being dissolved. So our takeaways here, we show that magnetofossil populations can be used to help piece together these larger scale environmental changes here at Wilson Lake over the PETM, and they have some advantages over some traditional proxies. All right, next I'm going to show you more of a bulk environmental magnetic approach to answer some of these environmental questions as well. We're still going to use magnetofossils, but we're going to use some other minerals as well. And this example is from Holland Ballpark in present-day Virginia. It's another marine sediment core that records a continental shelf environment. But what's different about this one is that it records Oceanic anoxic event 2, which happened during the Cenomanian Turonian about 94 million years ago. But we have another large-scale change in the carbon cycle and another analog for modern climate change. And you'll notice in the top figure here, particularly that it's in the center of 3 large igneous provinces. So we can begin to answer questions about sources of iron and the effects of these large igneous provinces on continental shelves. And just to place ourselves in time again, same figure going from 250 million years in the top left toward present and top right. Highlighted 94 million years ago was OAE2, Oceanic anoxic event 2. What's different about this event to the PDCM is that we have a positive carbon isotopic excursion, but we still have this increase in global temperature. Real quickly, here's the stratigraphy of Holland Park, and this is a ton of work done by my colleagues Jean and Christina at the USGS. But basically what they did, they used a combination of lithology and biostratigraphy to find the Cenomanian Turonian intervals on the far left. And particularly, they found these barren intervals represented by the dashed lines here, which we think is the local record and extent of OAE2 at Holland Park. Next, here are the results for total organic carbon and delta 13C carbon isotopes of organic carbon. And they both increase within that dashed interval that Jean and Christina came up with. But particularly, we see that it's 2 per mil positive carbon isotope excursion in our delta 13C. And I'm going to highlight that by the solid gray background because we think that that is the global record of OAE2 recorded at Holland Park. Next, here are some of our bulk magnetic measurements or results. So we have magnetic saturation, remnants, and squareness. And notably, where we see an increase in saturation and remnants, we also see this relative decrease in squareness. And we see this really nicely just before OAE2, and then again, just after OAE2, as recorded by the biostratigraphy. And then our last 3 columns here, we have magnetic coercivity, remnant coercivity, and our coercivity ratios. And similarly, where we have a decrease in coercivity and remnant coercivity, we have this increase in coercivity ratios. And again, it happens very noticeably just before and just after OAE2 as recorded by the biostratigraphy. So, as you may have picked up, we can start to split these into relative magnetic facies or bulk magnetic facies. And here to keep it simple, I've just done magnetic facies A and B. So facies A best describes the intervals both before and after OAE2, whereas facies B best describes the OAE2 interval. And my next question was, what exactly are the magnetic minerals that are making up these facies assemblages? So now we'll just take a quick look at magnetic facies A using this interval up there. And here's the resulting fork diagram for this magnetic facies. And hopefully by now, there's no argument here that this is single domain magnetite. We have this beautiful central red negative region, as well as a peak in the coercivity distribution at about 30 millitesla or so. We took it a step further and did some electron microscopy and found that there are both giant and conventional magnetofossils in this sample, which is personally very exciting because this is the oldest record of both conventional and giant magnetofossils, like physical visual evidence. But for environmental context, the presence of magnetofossils tells us that we have microaerobic bottom waters, and we at least had some sort of stratification happening during this interval. All right, taking a step back, let's look at bulk magnetic facies B using this interval up here. Maybe. All right, here's the resulting fork diagram. And this particular diagram has a very high central coercivity central ridge paired with this bean-like lobe and negative region below the central ridge. And you'll notice in the coercivity distribution, there's not a nice tight contribution to be seen. So all of these signatures taken together tell us that we have hematite in these intervals. And for this mineral to be preserved, again, as we discussed yesterday, the environment must have been oxygenated. So we have some sort of change in the hydrology, redox conditions, and iron cycling happening, perhaps an increase in runoff and mixing, or maybe even dust supply. Stay tuned. All right, so to sum up here, let's look at some of the environmental changes we can start to piece together at Holland Park. So firstly, we see changes in the iron cycle, just very generally. We have these dominant assemblages of single domain magnetite and hematite. We also see changes in redox conditions and hydrology, notably going back and forth between microaerobic and oxygenated bottom waters. And we also can study changes in the biology. So we see that these calcareous nanofossils and magnetofossils are not happy over OAE2. They plummet, but they recover up section. So we can start to study the ecological threshold of these organisms by looking at these records. So my takeaway here, we show that magnetic mineral assemblages can inform us about large-scale changes in iron cycle, redox conditions, hydrology over OAE2 at Holland Park. And we also show the oldest robust record of magnetofossils, highlighting the potential for even more of these critters in the rock record. All right, so today I showed you two examples of how we can use environmental magnetism to start to piece together these large-scale environmental changes that happen over these global warming events, particularly iron cycle, redox conditions, hydrology, nutrient conditions, and organic matter. So I just want to put a plug in there that environmental magnetism has a lot of advantages over some other more invasive techniques. But I would like to leave you today with some of the future directions I'd like to see, particularly magnetofossils going in. And of course, this includes better magnetic measurements that can get better quantitative results on these magnetofossil assemblages. So the 4PCA results were relative proportions of magnetofossils, but can we get actual numbers on them? And I think this is particularly important with disparity, so the differences in shapes again, as well as abundance, and with us finding even older giant magnetofossil assemblages. This also highlights the need to investigate these critters further and their contributions to the iron cycle. And Matthew Moore has started doing that, so just want to give him a shout out. But we definitely have a long road ahead of us. And I think some more ways that we can tackle this include modern observations. So after Allie's talk, I was like, I need to talk to Allie about this. Where we can actually get physical numbers to match with magnetotoxic bacteria populations. And of course, coupling this with micromagnetic modeling to better inform our simulations. Thank you very much.
All right.
Time.
No, it's a really dumb question.
No, I have no dumb questions.
What Ali was talking about in terms of the iron cycling, it seems to me that, and maybe this is a question for her. She was also talking about relative abundance, and you were talking about relative abundance, and it seems to me this poses something of a challenge in terms of real quantification. Is that right?
Yeah, unless two relatives make. Yeah, but I think at least when we're working with modern sediments, we might have a better chance at getting more quantitative estimates. But I think Allie and I need to talk about that.
So the path forward is really to focus on the modern sediments and then use that as a method for quantification so that you can go back and convince people you know what's going on in the past.
I think it's low-hanging fruit in like the best way, because especially with magnetofossils, we can visually see the bacteria and like measure that in real time. But I don't want to limit and say that we just need to do modern studies. We also need to. Yeah, thanks, Kathy.
Maybe a question about following on from what Kathy asked, going from relative abundances into absolute quantification that you mentioned.
Yeah.
So given that your three N members are all magnetofossils, what's stopping you from just multiplying those relative abundances by the SIRN? And then you know it's single domain magnetite. So you know you can therefore reconstruct how much single domain magnetite you have per N member. And you know the approximate sizes of the magnetofossils. So would that then take you into quantification?
Yes, and we did do a little bit of that in our paper, where we did do the math and got some estimates. However, I think it would be tough because I'm sure there are other magnetic minerals, other forms of magnetite in the samples. So I still am not sure if I would trust that to be like a perfect quantification of it.
Okay, so maybe an extra N member in there that you're not seeing in that case.
I feel like there's always extra N numbers in there. These are the dominant ones. Yeah.
Giants.
Yeah.
It's now kind of accepted to actually be magnetofossils rather than being produced by an inorganic process.
Well, Zhu actually came out recently with a nice paper showing how the structure of them really should be biological. But you know, the skeptic in me as a scientist is like, you'd always have to have that caveat until we actually find the critters that make them. But the shapes have not been reproduced that we can find like inorganically. So they really do seem to be biologically controlled in that sense. Yeah, great question though.
Thanks for your. I have a question about the giant magnetofossils. You have them done as being single domain, whereas there's a paper by Chang et al. 2012 where they showed that often they have vortex structures and multi-domain structures. I mean,
Yeah.
have you tried to accommodate? Sorry, I haven't read your paper. Have you tried to accommodate any of that in your method in your studies?
So yes, that paper by Chang was awesome. I want to highlight that was micromagnetic simulations of hypothetical arrangements of magnetofossils. So we still don't know for sure until we actually see how they're arranged and preserved in the rock record. But you're totally right. Many of the giant magnetofossils are likely to have single vortex and multi-domain signatures. And because of that, we don't pick that up in the 4 PCA yet. Maybe we can with like further development of it. That's again why we're so excited about the biogenic needles, is because they are a single domain even without having a specific arrangement in the sediment. So that's as much as I could tell you, I think.
Yeah, we have a few questions online.
Cool.
When Williams was asking a similar question to Adrian's, I noticed that you have non-rod type giant magnetofossils that probably won't have high coercivity.
Yeah.
Could these exist unnoticed in parts of your record?
Definitely.
Rich Harrison is making an observation that there's an absolute button in Fort EM that actually does try to help quantify some of the magnetization that's being observed.
I'll write that down. Thanks, Rich.
Adam is saying, do you have any available records of giant magnetofossils from quaternary marine sediments?
Do we?
That's the question.
Not that I know of, but they could be there. Let's look for them.
Can I ask one additional question about your second case study?
Yeah.
You had magnetic facies A and B.
Yeah.
And did you have any measurements from within the actual ocean anoxic event itself? It looked like all of your A and B samples were above it. Did I misread that I may have.
Do you just mean like for the fork data sets? Because all of that was Sorry? Oh,
Can you? Yeah, for the second case study, where they had magnetic phase sheets A and B, it looked like, her two fork samples were above the actual Oceanic amount. And I'm wondering if you had any samples from within.
so just to repeat for the people online, Josh is asking if we have fork measurements for the actual OAE2 interval and below, because I only showed examples from stratigraphic horizons above OAE2. And yes, we do. I just, yeah. I just chose these particular two, so I wasn't in a rush to make the talk, but yeah. Good observation.
Courtney, just a comment. If Joe Kirschvink was here, he would probably say, look at the 2008 Schumann et al. Paper in PNAS, look at the supplement. You commented that we don't know the arrangement of these magnetic fossils.
Are you talking about the Death Star?
Yeah. So, you know, they've got these X-ray images.
Yeah.
In situ with, as you say, a Death Star arrangement of these magnetites. Magnetotaxis seems highly unlikely. Given that arrangement, but yeah, that's the best evidence of in situ. So then what I would say is what you've been doing is extracting. That's what we did. But looking at in situ sediment imaging, there's Richard Harrison's online. He can tell you all about focused ion beam slicing view.
It's another thing we've been working on.
Yeah, I think those are methods that need to be applied to understand how these magnetites are arranged, what their potential function is for the organisms. If they are organisms that produce them.
Yeah, absolutely right. Yeah, thanks for that comment, Andrew. And with respect to the Death Star, we have great names for all these things, don't we? I actually thought that was loosely magnetically extracted, but because I know exactly what you're talking about. I believe those are also all spearheads. So with those, it actually, we wonder if maybe they're formed for some sort of like protection on a critter, because they all seem to be pointing outward too. So the verdict is not out on the different types of critters that made these and why they were making them. But I hope we find out. It's really cool.
And then one more question for Roger, and then we'll break for coffee. If anybody has any questions after this one, there's the coffee break, or we've got 15 minutes at the end of the session for all speakers.
Just a quick question, I guess. Andy actually covered a lot of it. So do you sense just how much the arrangement of the particles actually skews your coercivity distribution, right? Like you have these three end numbers individually, makes a lot of sense. Each one has higher coercivity. Right? But if one is arranged, in chains and the other is arranged in balls, right? How much do you have a sense like how much does that skew your distribution?
I don't have a quantitative number on it, but you're exactly right for noticing that it definitely does affect why we don't have perfect distinguishable end numbers, and there is that overlap because of that difference in not only chain arrangement but also the slightly different morphologies that make up those end numbers. So you're exactly right. They definitely are skewed because of that.
Well, thank you, everybody. 20-minute break now before we have a final two talks in the session. So